Okay, to start, we need to, what is a wild type non-mutated gene of sequence above? To know this, we need to copy this sequence, search in BLAST, NCBI, nucleotides, paste it here, run BLAST. After a few seconds, it will give me a list of results. The first one is probably the most similar one. Here I can unselect all and select the first one to download its aligned sequence, not the complete sequence. The complete sequence is the entire Escherichia coli sequence. Here we can rename it aligned, save. This is the first question. Second question, I define the most closely related gene species strain number. The most closely related is Escherichia coli. Its gene name, it is Escherichia. The species is coli. The strain starts from here and ends from here. That's it. Next question, determines the single number SMPs present in the sequence. To know this, we need to click alignments and minus these two numbers, which is 479 minus 469 equals, I rename it known, 478 minus 469 equals 9 single nucleotide polymorphism. Uh, present in here. We detect the pattern within the sequence. To know this, we can copy this sequence and use Eugene to find any pattern. We can name it a mutation. Great. To find any pattern, action, analyze, find pattern. ATG, for example, we have 16 occurrences of ATG in this entire sequence. Okay, then the next question, and we need to find the presence of tandem repeats. To find it, we need to click action, analyze, tandem repeat, find tandem repeats, whole sequence, start. There is one occurrence of tandem repeat right here. Okay, next we need to design primers inclusive of the start and stop codons. To do this, we can select all the ORF site and press analyze primer 3 plus, check left and right primer. General setting, the thermodynamics parameter is this. This formula is what we'll use, and this is the selected region. Pick primers. Here I have picked already the primers. To view them or to analyze them, we can press this circular button to analyze it right like this. This is the position. To integrate BAM-H1 restriction site, we can copy it. We can go to Google, search for BAM-H1 restriction site. Here I can copy BAM-H1 restriction site from here, for example, and I can remove this wrong part, copy it, and integrate it into the designed primers. To do this, let's close this circular view. This is the primer that I want to integrate it. Edit, switch on editing mode, and place it just before the primer. If this is the primer, this is the second primer, then I will place it right here to show the restriction site. BAM H1 right here and right here. Then the program have recognized this restriction site. Then clone the genetic modified gene into POC19 plasmid vector. To clone POC19 plasmid vector, we need to go to NCBI database, CBI database, POC19 vector, nucleotides database, FASTA format, and copy this sequence. Then go to Eugene, start page, create, paste this sequence, then rename it POC19, save, create, circular view. Here is a POC19 plasmid vector. To insert our gene, we press action, cloning, digest into fragments. We digest with BAM H1, okay. Here we have two fragments. To actually clone it, we need to construct a molecule with POC19 first fragment, and from our project, the mutation, and press OK, and force blunt make circular. Okay, here is the POC19, and here is the mutations that I have inserted. Then we need to identify 1000 base pair segment located upstream and 10 kb upstream from start codon. To know this, we need to go actually to the first blast search that we did to download the complete sequence of the organism, Escherichia coli. Let's name it complete. And from start page, press open files and open the complete sequence. Here, to know our exact location, we can go to alignment. This is the exact location of the gene. Select region. We can place it right here and right here. This is the exact start of the gene. We need to go 10,000 before it. So we need to go actually a lot further up. Let's actually search it again to bring us to the beginning. Here it is the beginning. Then we need to go 1000 base pair up, just like so. Here is the 1000 base pair. That's it. This is the ninth question. Predict ORF size within the 100 base pair sequence, ensuring minimum ORF size of 150. We need to right click, analyze, find ORF within the selected region, 150 for the minimum, and press OK. Here I have just found one ORF within the selected region. Next, we need to generate protein sequence with AlphaFold 3 and analyze the 3D structure. For it, we can search for biomodel protein sequence, copy our mutated gene, place it here, transcription, translation. Here's our mutated gene. We can actually go for AlphaFold to Google Colab. Go first link. And place our amino acid sequence here. Then run time, run all. Run anyway. After a little bit of time, let it run. Then we will find that it had made me a 3D structure, like so. And it have actually downloaded the PDB structure right here. So that is this question. This is 3D structure. To conduct comparative analysis between this and the original wild type gene, we actually need to go to UniProt, specifically BLAST UniProt, and run BLAST on my mutated gene to know the gene name. Run BLAST. After a little bit of while, it have actually finished, and here is the result with the ACCB gene name and this protein name of my mutated sequence. So what actually you need to do is to compare this two 3D structures. So we need to search PyMol on this PC, and download the wild type. The wild type, we can actually find it in the AlphaFold database by searching this specific protein name. And we can press Escherichia coli because it's the algorithm that we have, and download its PDB file. Okay, we have downloaded the PDB file. Let's name it from alf because we got it from AlphaFold. Save. Then we need to combine those two by unzipping this file, extracting here. Let's name this the mutated, and here we place the mutated in the bimol and the alpha PDB right here. Then we can do alignment by align the mutated and the alpha1. Alpha1, yes. Here it actually showed me a RMSD value of above 1, which means that the mutation was so severe that it actually changed the structure, the physical uh, structure of it. That means its function is completely different from that it was before. So this answers this question and this question. Shows the gene pathway pyruvate metabolism in KEGG. We can actually go to KEGG right here and search the gene name. The gene name we actually got from here, uniprot bast ACCB. We can search it right here. Search. First result. Pyruvate metabolism. And here it is. After a while it will highlight red. Yes, because it's mutated, it will affect the acetyl CoA from transforming into malonyl CoA through this enzyme. So it will affect all these other pathways in the INCAG. So next we need to identify three genes affected by the rank in string. So we need to go to actually to string database, string database here, and search the protein name, which is this. Remove any excess information and press search. Okay, still remove this, press search. Here we have Escherichia coli. Let's continue. Here ACCB gene, and because it's mutated it will affect all these other genes, but it's dependent on the number of strings attached to it. Here we have 1 2 3 4, so this is 4 strings, so it's affected by 4, and this is more, so we can arrange them like this, and this is just 3, so it can be arranged like this. More analysis can be done to accurately arrange them together. And this is the final question, and thank you.